Welcome to my presentation on lipid therapies in mouse models of Pelizaeus-Merzbacher disease. I'm Gesine Saher and work at the Max Planck Institute of Experimental Medicine. First, I would like to thank the organizers to invite me here for this presentation, and I'd like also to welcome the PMD families and the scientists. This is my group, here located at the Max Planck Institute of Experimental Medicine in Göttingen, which is in Germany. And this is my group we work on cholesterol in the nervous system, and Sina Stumpf here acquired basically all the data that I will present today. Let me start by giving some introduction on myelin membranes and oligodendrocytes. In the central nervous system, oligodendrocytes extend processes from the cell membrane that wrap around axons. And then after a process called compaction, these processes build up a multi-layered stack of membranes that you can see here in the dark ring around this axon. In a high-power view, you see the stack of membranes quite nicely, and here in this cartoon you see that these membranes contain myelin proteins, proteolipid protein PLP, of which you're all aware of, and also lipids. One lipid of this membrane is very important, and this is cholesterol. 70% of brain cholesterol resides in myelin membranes. And another aspect which is very important here is that PLP directly associates with cholesterol in myelin. What is myelin good for? It insulates axons, and therefore also the lipid-rich constitution is very important because this insulation function relies on the lipids. This allows saltatory, so fast impulse conduction, here over the internodes. Another feature of myelin is that it reduces the energy demand of axons to maintain this impulse conduction by about 80%. This has been calculated. To give you a little bit introduction on the cell biology of the process of myelination in normal wild-type oligodendrocytes, PLP and cholesterol associated within the cell. They are transported to the cell membrane, and upon myelination, this process is increased and leads to the formation of myelin membranes. And you can see in this micrograph of a brain section of normal mice, PLP here in green, that myelin is all over the place in the white matter. However, in a PMD mouse model, the picture is different, and the green structures here are located around the blue stained cell nuclei. In the cartoon, this in classical PMD, which is caused by an overexpression of PLP protein due to the gene duplication, we see that more PLP is built within the oligodendrocytes, and this alters the ratio of cholesterol to PLP in the cell membrane. Upon myelination, this leads to the accumulation of PLP and cholesterol within the cell, and this impairs intracellular transport to the myelin sheath and hence poor myelination. We addressed this feature here of the altered ratio of cholesterol to PLP by adding extra cholesterol to the mutant cells by feeding a cholesterol diet. This normalized the PLP to cholesterol ratio within the cell membrane, and the accumulation intracellularly was relieved by this treatment, and this led to enhanced myelination. I want to stress, however, that we did not correct the PLP overexpression. You see, this overexpression is unchanged, and this part of the PMD pathology is addressed in gene suppression therapeutic approaches, and you have an own session about this. But when we treated our PMD mice with cholesterol, we found that the myelination was almost normalized. This was very, very remarkable. But how does this cholesterol really reaches the myelin sheath and the oligodendrocytes? If you have the brain, then of course you need the entire brain vasculature to transport components of the diet towards the brain. But here is a limitation in the transport of external substances to the brain for good reasons. And this is the blood-brain barrier. The endothelial cells of brain capillaries prevent the import of many substances into the brain, and cholesterol, unfortunately, is also not transported. So this treatment benefit of cholesterol in our mice was achieved because these mice have a leaky blood-brain barrier. This allowed cholesterol import and improved myelination. We have another PMD mouse model with a tight blood-brain barrier, just an almost normal blood-brain barrier. Here, no cholesterol import occurred, and we also see minor benefits on myelination. This was unfortunate for the mice, and also in the 2 PMD patients that were treated with cholesterol did only show minor benefits of the cholesterol supplementation. But coming back to the pathogenesis in PMD, it is well known for a very, very long time that problems with myelination sooner or later lead to some axon damage, which sooner or later leads to some neuron loss and brain atrophy. But why is this so? We hypothesize that energy deficits in axons and neurons contribute to this axonal damage, because I told you that to maintain impulse conduction without myelin, neurons need about 5-fold more energy to achieve this. So what does people or things people do when they experience an energy deficit? They increase power stations. The power station of the cell are mitochondria. This is here an electron micrograph and a cartoon of how these mitochondria look like. And if one senses energy deficits, mitochondrial size and density will increase. So we first tested whether this is also the case in our PMD mouse models. And indeed, here in wild-type mice, at 3 weeks of age, we see some mitochondria, you can see them here, and they're small and scarce. However, in our PMD model, we see that the sizes of mitochondria have increased, and also the numbers of mitochondria here are increased, so implying that really also in these PMD mice, where the genetic defect is only in the oligodendrocyte, not in the axons experience energy deficits. This increase in the power stations of the cell come also with some problems, because it increases the physiological stress and might also increase the amount of reactive oxygen species and other reactive compounds. And this part of the PMD pathology is addressed by treating models and maybe also now patients with chelators of iron, which are devastating when they are reactive, and also just normal antioxidant treatment, which has been done in cells. And I think Nicole Wolf will give you some more details about that. But our hypothesis was that especially lipids are important or can have very beneficial effects on the PMD pathology. So we thought about a new lipid treatment approach. Lipids do often react as oxygen scavengers. They can provide metabolic support for axons, and the treatment should be efficient, independent of blood-brain barrier disturbances. So we came then to the idea to treat our mice with a ketogenic diet. What is this? A ketogenic diet is a diet which is very high in fatty acids or in fats, and very low in carbohydrates. It's a high-fat, low-carb diet. This diet works such that the fatty acids, or the fat from the diet, is converted in ketone bodies within the liver. They are then released into the bloodstream, and you can measure this here by enhanced serum levels of beta-hydroxybutyrate. These compounds are now taken up by the brain, and they are converted to energy in a process called ketone body utilization, and the protein OXCT1 is a rate-limiting protein in this process. In our PMD mice fatty ketogenic diet, we found that OXCT1 levels were highly increased in comparison to a standard diet fat mice, and this of course implies that the ketone bodies are really used in these mice, in the brain of these mice. What effects does this have on the pathology of PMD? We first looked for oligodendrocyte numbers and found that the loss which occurs in untreated PMD mice was rescued by feeding a ketogenic diet, and also myelination improved a little bit. When we tested motor function here with an elevated beam test, we found that here in green, the mice on ketogenic diet were behaving like wild types. The somewhat smaller benefit on myelination could not explain this increase or this better performance in this motor test. So we specifically looked on the well-being of axons and stained axons for axon damage, which is here APP. And we found that the abundance of APP spheroids was strongly reduced in PMD mice fatty ketogenic diet, implying that really the axon damage was attenuated. I told you about the increased size of mitochondria in axons of PMD mice, and we found this here also in later ages. And when we looked for mitochondria size in PMD mice fatty ketogenic diet, we found that this was basically normalized. And this is also seen here in the 3D representation of mitochondria. The next thing, of course, is whether also the impulse conduction ameliorated. And indeed, here I won't go into details of this electrophysiological analysis. Yes, we did see improvements in impulse conduction. To summarize now my talk, we found that myelin disease in PMD is associated with mitochondrial defects in axons, which quite likely lead to axon damage and persistent disability. When we feed mice a ketogenic diet, this improves the pathology of PMD independent of blood-brain barrier disturbances. It reduces ER stress, which I haven't shown you today, but it also enhances oligodendrocyte numbers and it supports myelination. More importantly, ketogenic diet reduces energy deficits in axons and ameliorates disturbed impulse conduction, which improves the clinical outcome in our mouse models of PMD. So to summarize, while the cholesterol mainly supports myelination, the ketone body treatment supports the well-being of axons. And with this, I'd like to thank not only my group, but also the other collaborators and in our department here of Klaus Arvin Narve in the Max Planck Institute of Experimental Medicine, but also our clinical collaborations in Hamburg and in Göttingen. And I thank you for your attention, and I'm happy to take any questions. Thank you.